So, let's get started. So today I'm technically gonna teach the part circulatory system or basically heart. Okay, so it's according to the ICSE syllabus. So I'm gonna go the way it goes. Starting one is composition of blood, structure and function of it, RBCs, WBCs, and platelets. So something you have to remember is that in our body, let me write down the title heart. So technically in our body, the whatever our body has three main fluids. Okay, the three main fluids are as follows. The first one is blood, the second one is tissue fluid, the third one is lymph. So these are the three main fluids. uh, so let's start off with talking with blood. uh, so the main the okay, let me write it down somewhere here. Write down blood. Its main function, its two main functions. Uh, um, Wait, let me write function. Its two main functions is to protect, okay, and transport. So first of all, it's gonna be transport and then protect, of course. So transport what? Of course, we have all type of uh, digested food. Okay, let me just write down here food and the transport of oxygen, carbon dioxide, technically all uh, gaseous elements that our body needs. Example for this is of course O2, CO2, etc. And it's transport of excretory material, excretory. So if we have something like XO8, mm, there's no E there. Excretory materials, metabolic waste and all that stuff. A distribution of hormones and distribution of heat. So it also distributes heat. These are common stuff you can go through. It also, how does it protect? It forms clots. So formation of clots are one of the main thing here. Now with all that stuff, let's see. Now composition of blood. Let's look at its composition. So blood, or technically RBCs, it's mainly composed of, here is our test tube. So RBCs is mainly composed of the red blood cells and another layer called the plasma. Okay, so it's technically made up of plasma and RBCs. I mean the what solid matter, you can tell, the solid matter or the cellular element, something like that. Um, it also contains a few inorganic salts and substances like glucose. The plasma contains substances like glucose, amino acids, fibrinogen, hormones, urea, etc. etc. So one thing you have to remember is the plasma from which protein. So if from the plasma, if protein if protein fibrinogen is removed, fibrinogen is removed, it will form something called serum. Yeah, hope that is clear. That is one of the important things. So in the cellular elements of plasma, okay, in the cellular elements, let me write that down. Cellular elements, that is technically this thing. The cellular elements mainly composes of uh, three main things. Let me write that down here to make it easier. The first one is WBC, white blood cells. I don't have to explain that, do I? These are also called the leukocytes. Leukocytes, the RBCs, which is erythrocytes and blood platelets, thrombocytes. Um, Thrombocytes, okay, I mean platelets. So let's start off with WBCs here because it's a bit longer than everything else here. Okay. WBC. So the WBCs, let me first go to the WBCs. Where is the WBCs? Yes. So WBCs are leukocytes. So these are white blood cells. They are technically found in between RBCs, and they are way much larger than RBCs are. Okay, or are they smaller? They're much larger, from what I know. So WBCs are called the white blood cells. I mean, the abbreviated form is WBC. So mm, so this is in blood. Okay, in blood there's WBCs, huh? Okay, so these are white blood cells, and they are mainly of 5 types, which I'll come to. But there's something you have to remember. So there's a process called diapedesis. Okay. Oh shit. Huh. So there's a process called diapedesis, and there's a process called diapedesis, which is the oozing of WBCs. Oozing of WBCs out of the cell. They migrate outside the. You know what? They migrate outside the blood vessels, so as to when they find something like a foreign body, so they migrate outside of this thing. That process is called diapedesis, and this is something to remember. So let me highlight it. It is important. Okay, till now, from whatever I have told you, this is one of the important key points. It might come as this thing, like a one marker, so yeah, it's better to remember. Let me get back to that. So now diabetes is oozing of WBCs, and then the next thing we have is mm, phagocytis. Phagocytosis, sorry. Phagocytosis. Right there. Pencil is a bit. Okay. Phagocytosis is technically uh, WBCs engulfing the WBCs engulfing foreign particles. So engulfing foreign particles. Okay, and then a few Huh? There's also another process called inflammation. I mean, it's not a process. This technically happens when uh, when WBCs localize any injury or this thing. So it causes redness, swelling, pain. So this is followed by diabetes. So after diabetes, it's this. Okay, it causes this. So let me write it down here. What is it? Localize of germs. They're technically neutralizing anything that the germs have secreted. Um, they also destroy damaged cells, so that is something to remember. Destroy damaged cells. Uh, so this causes pus, red redness in the area, and swelling, pain, etc. This is something important to remember, these three. So let me get that down for you. This is also important to remember, so hope you'll certainly remember that. These are technically definitions, so you have to remember. There is also formation. So these are all these things that you have to remember, under which, okay, let me start this stuff. Star, that is another point. Inflammation is another point. And there is, so let me write it down somewhere for you guys. All of this is functions of WBC. Okay, and the last point is this the last point? Yes, the last point is formation of antibiotics. Forms antibiotics. So produce antibiotics which kill or neutralize germs. So it's technically what like inflammation. During inflammation, antibiotics are formed. This is mainly lymphocytes. We'll come to that. But so now that we have done with all the functions of WBCs, let me point that thing. That is functions. Let's go on to the next point, which is let's do a few diseases related to WCC. Diseases related. So two of these which you have to remember is leukemia, cancer of the tissue. So technically WBCs increase, but at the cost. But at the cost of RBCs. So technically, what happens is WBCs are increasing, but in the process, RBCs are being destroyed, and leukopenia is this can come as a one pointer. You have to better remember this. This is just increase in WBCs. Abnormal one though, you have to remember. Usually, WBCs will increase. The count of WBCs in your body will increase if there is any type of, what do we call it? If there is any type of increase in germs or any type of foreign body which increases, so Now let's come to types of WBCs. These are only the important points, which is like for a quick revision that you want. Um, so there are 5 types. Let's start off with the first one. Neutrophils. Neutrophils are like, these are the main ones. So there's like 55% to 70% of these. Let me try to replicate the diagram. I'm sorry, I'm bad at drawing. So they're amoeboid in shape. You have to remember that it's not like a proper. These are 3-lobed structures, so it has 3 lobes inside them. And there's some weird particles in that. I'm trying to replicate it. I'm sorry, I'm so bad at this. So let's go back to the points. So again, the noticeable thing is 3 to 4 lobes. They have granular cytoplasm. So these both are depicted in here. So the cytoplasm is granular, and then 3 to 4 lobes. I just mark it that this is the purple shades, and that is the pink shade. Okay, once that is done, the function is to engulf bacteria. I'm going to do the second one a bit up here, just so that I use up all the space, because I don't like wasting space. Second type is eosinophils. Eosinophils, you know, yeah, eosinophils. Now, this is pretty low. I don't recommend you have to. I don't think you have to by heart the percentage here. Okay, so let me draw the diagram again. These are 2-lobed structures, that is, they again have granular cytoplasm. Is that it? Large cytoplasmic granular, yes. The cytoplasmic granular is large. Okay, so main points from here: 2 lobes, nucleus is 2 lobes, and then another point is a large granular cytoplasm. Two points to remember: they also called eosin because eosin is a red dye and they get stained under it. Okay, and eosin is not red dye, it's a dye which technically turns this red. Dye turns it red. Eosin is the dye. Let me highlight that eosin is a dye. Then decrease that size of it. Okay, eosin is a dye. Okay, and that's why hence its name eosinophils. Its function here is engulf bacteria. It's fine, it engulfs bacteria and all that. This is mainly phagocytosis. If you remember what phagocytosis is, I forgot to add in here. This is the main substance, mainly neutrophils. Okay, phagocytosis. is the engulfing of bacteria. So mainly neutrophils do that. And here we have, so the function for this is, um, the function of eosinophils is mainly it is associated with, this is one of the main functions. It also engulfs bacteria, but this is one of the main functions, and I wouldn't recommend to read the other functions because you might get confused. So allergies, this is the cause. If there's any type of this count increases in allergies. So if they ask you what count increases in allergies, you are gonna mark this. Okay, if the what type of WBC's count increases when there is an allergy, it's eosinophils. uh, by the way, I've forgotten to tell this, but I don't think it's required. It's not required. Okay, I'll tell it anyways. The production of neutrophils, eosinophils, and the next one, which I'm going to tell, basophils, are all in bone marrow. Everything is bone marrow, so you can just tell bone marrow for everything. I won't tell much of it. I'll just write it on top if you want. All produced in bone marrow. That's easy. Better remember. I don't think they're gonna ask it, but fine. The third one is basophils. This is again very low. Don't need to remember the percentage for this. It's like 0.5 to 1%. Doesn't matter much. So let me draw the diagram for you. Okay. There is this very large, indistinctively lobed nucleus here. And so we are okay. So this is indistinctively indistinctively lobed, and is again granular cytoplasm. All of these three are granular cytoplasm because they come under the two. This technically it's divided into two, and there are two main types, which is granular WBCs and non-granular. So remember that all these three are granular cytoplasm, so all of these come under that. I could have written that at the start, but fine. And the function is, so this releases basophils. They release chemicals for inflammation. So anything related to inflammation, it's, basophils. That is this. And then the fourth and fifth one we have it. I'm gonna write. Okay. Yeah. The fourth one is, after this, the fourth one is lymphocytes. Here's a bit change. So this is a non-granular one. Okay. Non-granular, and this is smallest. I'm gonna draw it for you. Wait. I'm gonna draw this relatively small so that you can make it. The nucleus is very big, and that's the main points here. smallest, one large nucleus. And the function is to produce antibodies. That's it. That's all you have to remember. And this, along with the bone marrow, it is also in lymph glands. But I wouldn't, I wouldn't sit and read that because I don't think it's gonna come. The fifth one, but it's the last one. Monocytes. Oh wait, yeah. So this lymphocytes is 20 to 35%. So that's relatively high. So remember that this is the second lot. And monocytes, there is 3 to 8%, doesn't matter. Let me draw that out. Oh yeah, these ones are the largest, rather large, and their nucleus is kidney shaped. Yay! I'm not gonna sit and fill that up. Let me increase that and let's fill this up. They have kidney shape. Kidneys, big fat kidneys. Okay. So the points are here. Largest, these are kidney shaped nucleus. Kidney nucleus, and the function is ingest germs. So that's all. Let me highlight the function so that you can go through it once. In neutrophils, they have 3 lobes: granular cytoplasm, these engulf bacteria, that is phagocytosis. Eosinophils, these are 2 lobes, large granular cytoplasm, and associated with allergies. The next one is basophils. These are indistinctively lobed granular cytoplasm, and they release chemicals for inflammation. Remember that. Now the second last is lymphocytes, 20 to 35%. Neutrophils is the most, lymphocytes is the second most. These are smallest, one large nucleus. Function is to produce antibody. And the last but not the least is monocytes. They are the largest. They have kidney shaped nucleus, and the function is to ingest germs. You can go through that once. And now let's continue on to the next one, which is, I should get better at drawing. Okay, after WBCs, the next cellular element we have in in blood along with plasma in cellular elements. The first, okay, let me get through this way. The blood contains plasma and cellular elements. Cellular elements are mainly divided into 3. Now the second one is, we are going to talk about RBCs. Okay, RBCs, let's get to these. Where was I? Let me go to RBCs. Okay. So in this, let's talk about the structure. Biconcave shape. They have a biconcave shape, and disc-like structure. So let me disc-like. So technically this is how it would look: a circle with a biconcave structure. I'm gonna fill that up here. I know this is a pink color, but this is for the aesthetics, okay? Just don't mind me. Um, Technically, it consists hemoglobin, which I'm pretty sure everyone knows, and for that you need Fe, that is iron. Just telling you if you don't know that. Now let's go back to the white part. That's for the structure. Now next point we have is hemoglobin. Hemoglobin, there's it's made up of hemin, which is a protein. It's iron containing part. So this is the Fe part, and globin, which is a protein. Those are the two things that make it up. Hemoglobin, it's technically oxyhemoglobin, two parts, which is, that is the term. The term can be divided or derived from those two words, and then there's oxyhemoglobin, globin, and carbaminohemoglobin. Carb amino hemoglobin. That is very long, okay? So something, this is, let me write down H, B, and oxygen. This is H, B, and CO2. That is hemoglobin with oxygen, hemoglobin with carbon dioxide. There is another case where hemoglobin has a strong affinity. Let me write it down. Strong affinity. It has a strong affinity for, you know, carbon monoxide. Okay, and since people smoke and all that stuff, if carbamino gets to this thing, like if so it's gonna end up forming HbCO if these two meet. So all the smokers and all, that's what it happens. So carbon monoxide mixes with the hemoglobin, and since hemoglobin has such a strong affinity for carbon monoxide, you know what will end up happening? It's gonna end up being stuck there, and there is no space for oxygen or carbon dioxide to take over. This is also called a carbon monoxide poisoning. Let me just read this part out because this might come as a key reason. Hemoglobin has a very strong affinity for carbon monoxide, forming a stable compound, carboxyhemoglobin. So there's a difference. Carbaminohemoglobin is different, carboxyhemoglobin is different. Remember that. I'm not going to write this down because it's going to confuse it. If you want to go through it, go through your textbook. This cuts down the capacity of blood of transporting oxygen, sometimes resulting in death. This is also known as carbon monoxide poisoning. So sleeping near burn furnaces in small rooms without ventilating may cause this, and even smoking causes this. Now, life and death of an RBCs, they are okay. Well, let me write down the production is in the production is the marrow of the long bones. So marrow of longer bones produces these RBCs. They mature. Now let's go back to the structure part. Back on cave shape. Okay, let's write absence of structures. Organelles. Absence of organelles. That's a This is important because they might again ask you questions on this, give reasons technically. So, due to the first, let's get to know what and all is absent. So, in a hemoglobin, okay, in RBCs, in mammalian red blood cells to be specific, there is no nucleus, no mitochondria, no endoplasmic reticulum. So, let's start off with nucleus. Now, I'm going to give you the reasons here about why there is no nucleus. Makes the red cells, it makes it bind concave. Okay, which in turn makes more area, better absorption. I'm sorry for the spellings, don't go. Okay, better absorption of O2, oxygen, better absorption of O2. So, what makes it technically makes it biconcave in shape. So, let me draw that. It's like this from a side view. Oh shit, it's like this from a side view, and from the frontal view, of course, it's like that. Yeah, that is how it looks. So, from the side views and the frontal view, better absorption of O2. That's this thing. Now, the second one is non mitochondria. No mitochondria, technically, cellular respiration occurs in mitochondria, thus all oxygen absorbed from the lungs is transported and delivered to the tissue. So, red blood cells, so this is technically where respiration occurs, okay? This is a bit hard to understand, so let me tell you properly. Respiration occurs in the mitochondria, and absence of it prevents RBCs using O2 up. So if there is no mitochondria, there is no cellular respiration happening, so RBCs will not use it up. That's technically one of the this thing. And so all the oxygen absorbed from the lungs is transported and delivered to the tissues. Last mitochondria means full transport of glucose in, okay, and mitochondria takes up energy, which is, you can write it down, but I'm not going to write that down because that's not the main. And then it does not have endoplasmic reticulum, or ER. I'm writing that, reticulum, yeah. No endoplasmic reticulum is more flexibility, so flexibility and easier movement through capillaries. Yay, we did this. Let me highlight all the three: absence of organelles, nucleus, mitochondria, endoplasmic reticulum. Let me do this highlighting too, because why not? Now, once that is done, oh, that's much of it done. So, RBCs, okay, I have not written this down. These are called leukocytes. These are also called erythrocytes. Remember this because it is important. I forgot something. We have a third part. I'll come to that. There is one more thing which technically I'm gonna write it down here very fast. Increase in RBCs will cause polycythemia. I don't know how to spell this. Polycythemia. And decrease in RBCs will cause erythropenia. That's much of the RBCs done. Well, there is another part which is thrombocytes. I hate them. I don't hate this, but I hate the lymph part, because I myself had a hard time understanding. So this is very bad. Let me. Okay, the cellular element third part I'm gonna do here because I'm out of space. Let me draw lines to cut it out. No. Let me Let me draw lines. I'm so sorry for this. And thrombocytes, let's do. Okay, the last part but not the least, because this is the worst. Okay, I hate this and I don't like understanding this. This is a bit longer, but we'll get through this. Okay, the last part but not the least is thrombocytes, of course. Thrombocytes, let me write that down. Oh no, what did I do? Okay, yeah. thrombocytes, thrombocytes. So thrombocytes are, quote unquote platelets. So they are formed. So they are formed in some giant cells called megakaryocytes in the red bone marrow. And destroyed in spleen. So they are formed in red bone marrow, destroyed in spleen, all that stuff. So the main function their main function is to initiate blood clotting. Okay, initiate blood clotting. And okay, I'm gonna explain this very as easy as I can, so that, because in the book it is explained very you know what intensely or very detailed. I'm going to explain it very lightly so that you can understand, and it's easier to by heart it. So let's talk about coagulation, that is clotting. So what happens in the blood clot is there is an injury in the area, something like that. Let's make it bloody, and it goes from the injury. When there is an injury, there is release of what, I'm gonna come to that. There is release of thrombokinase. thrombokinase, thromboplastin. This goes by many names. I mean, it calls Stewart factor, X factor, all that stuff. But they might give you these two. Plastin. Okay. And once that is released, along with uh, what this does is it acts like a catalyst. So it turns along with Ca plus, so Ca calcium ions. Am I correct? Let me just check that. So it converts to prothrombin to thrombin. So prothrombin to thrombin. Let me have that. Prothrombin to thrombin, and this with the help of potassium. I mean, vitamin K. Sorry, why am I this? I'm gonna write it as Vita K. Hopefully, you understand. Vitamin K. Mm, okay. Oh, sorry. Here, vitamin K is also required. I'm sorry. Vitamin K and calcium prothrombin. So, vitamin K is required for prothrombin, and prothrombin to thrombin and thrombin in the presence of Ca plus again will turn the fibrinogen, which is soluble, to fibrin. This soluble, this insoluble, and this fibrin, uh, you know, it is like a thready structure. Okay, it's like it forms like a thread-like structure, and these thread structures are very sticky. This sticky threads. And what actually happens here is these sticky threads, they bring all the cellular matter together and press out this thing. So the serum is formed. Let me do it in a diagram form. So these bring out all the, I mean, they bind all the RBCs and all the cellular matter stuff, and at last what we are left is these sticky threads. These are cellular matters, and the leftover part is called serum. So they stick to each other, they form a type of clot, and then the leftover part is serum. It happens like that. Okay, hopefully you understood this. I really can't help because not that great at explaining this. We have done with all the three parts. Now let's go back to another topic under this heart. Let me add a line here. So the next part we are gonna discuss is ABO and the RH system. EBO and RH. ABO and RH system. ABO and the RH system. So Okay, one more part. I'm so sorry. I'm so sorry for this. I'll write it on top here. So, diseases related to it are hemophilia, where blood cannot be clot. I'm pretty sure you guys know what this is by now. Hemophilia, philia, and dengue. You guys know what this is, so I'm not gonna explain it. Yeah, I'm so sorry for that. ABO and the RH system. This is not fun. It's boring because it's it's technically repetition of what we have. So this technically there is a guy. Okay, I'm gonna explain this as fast as I can. Carl Landstiner. His name is Landstiner. Carl Landstiner, and he he finds specific stuff. Okay, he finds something on the RBCs. On the RBCs, when he was looking at on the surface of the RBCs, he finds oh sheesh, he finds he finds stuff on the RBCs. Okay, and then he goes on to name proteins these proteins as antigens. It's not antigens, it's antigens. And he also finds something like that. Let's assume this white thing is our plasma, along with along with RBCs. Along with RBCs and all the cellular stuff that he finds, he also finds something. Let's take green. What? He also finds other things, and in the present in the plasma. These are complementary proteins. Proteins. These are complementary to the antigen proteins, and he named these antibodies. Get back to my old color. Okay. So technically, based on this, he named all of this. Okay. So I'm just gonna do a quick part for this. I'm not gonna go through all of this, but then just a small quick revision for you guys, because you must know this. I mean, who doesn't know? Act as if this is a table. Okay. So this is so there are different types of blood groups based on this. He finds two antigens, that is A, B, and corresponding to this he finds A, B. So a person having A antigen will have. I'll come to that. Let's talk about this. So we'll write antigen, antibody. I'm telling you complementary. So if a person has antigen A, they'll have antibody B. If they have antigen B, antibody is A. If they have no antigen, then they'll both have A and B antibodies. If they have A, B antigens, there is no antibody. And based on that, they have given the name of A blood group, B blood group, this one is O blood group, and this one is A B blood group. Let me draw lines to make it a bit readable. Yay, the table is done. This is easy. And let us remember, this person is universal donor. I mean, not this, but oh, eh, I'm gonna suffer. Okay, let me draw these lines back fast. Yeah. So there was another factor which is known as the RH factor. RH factor came so because they first found this in the rhesus monkey. Came RH factor. That's where they took the RH from. So unoriginal. I know. I understand. This technically determines the plus or minus of this thing. If RH is present, then it's plus. If it's not present, then it's minus. So blood groups of donors and blood groups of recipients. This is very easy. Why am I even telling you guys this? I'm gonna write a small notepad here so that you guys just go through this. O negative is universal donor. AB blood type is universal recipient. That's it. I'm gonna box that note in so that you guys will notice it. Yeah, that's it for the ABO and RH split systems. Let me erase that. Let me draw proper lines this time. Yay. ABO blood system done. We are done. Just left with the blood circulatory system. Oh, systemic pulmonary. There is a lot to do. Okay, we have come a far way. We have to finish this. I mean, we have done a lot. We'll finish this. Oh, the heart. You know what? Give me a second and I'll put in a diagram of the heart. It is easier to understand that way. Okay, I'm back. I've done this. So, don't mind this. I know I didn't find a transparent background, but it's at least marked and all that arrows exist. I am going to go through this. I have to use another color for this. Let's use this blue. Yes. So I'm gonna tell you Now imagine this is our heart. We have an upper body here. Upper body, and then there is a lower body here. Lower body. Okay. Now, based on this, this upper body also has lungs in it. Okay. So let me start off. Hmm, I'm gonna use pink here. Okay. The blue one will show you all the, you know what? Yeah, wherever deoxygenated is, and the red pink one is oxygenated. Of course, remember that. So let's start off. Okay, it's called the heart. The human circulation system is called double circulation because there are two type of circulation. I mean, the blood passes to the heart twice, and then it takes two parts to complete one circulation. So let me just tell you. I'm going to tell you the root first, and then I'm going to explain you both the systems. So with it starts off in the right atrium. Don't forget this. It's the right atrium. Mm. So it starts off with the right atrium. So from where does it go? I'm gonna write down the pathway towards the left way. So superior to the left side. I'm gonna write it down in purple. Okay. It starts off in the superior. I mean, from all the upper body to the lower body. First, let's do the marking. From the upper body to the lower. I'll make it thicker too much. Yeah, from the upper body it goes to the superior vena cava, and from the lower body it goes to the inferior vena cava. So both these it comes from here and from here. Superior vena cava and inferior vena cava, both of them bring it into the right atrium here. From the right atrium it goes, when the right atrium contracts, it is pushed into the right ventricle, and when the right and so when the right atrium contracts, the right ventricle is, relaxed, and from the right ventricle it goes into the through the pulmonary valve. It goes to the heart by the pulmonary artery. I mean, it goes to the lungs by the pulmonary artery. So I should have explained the structures first. The heart is divided into four chambers. Let me tell you. I'm sorry. I should have explained that first. But now you know the root. So I'm explaining it along with the root. So we have superior vena cava and the inferior vena cava. These are the two. Veins which bring the blood into the heart. So something to remember is veins bring the blood into the heart, arteries away from the heart. I'm gonna write that down afterwards here. Uh, and then there's the right atrium. As you can see, heart has the four chambers, and it starts off with the right atrium, and that's where the deoxygenated blood goes. From the right atrium, it goes to the right ventricle, which is the lower half, and from the right ventricle, it goes into this artery, which is pulmonary artery, but it's weird. It's going away from the heart, veins towards the heart, arteries away from the heart. Wait. Okay, yeah, this is where you get confused. Even I got confused. So veins are bringing it towards the heart. The arteries. the arteries are always away from the heart. Yeah, that's correct. So even if it's pulmonary artery or even if it's pulmonary vein, they follow the same rule. Veins towards the heart, arteries away from the heart, arteries away, veins towards. But the only difference is that unlike the other arteries, this artery, as you can see, this artery here, it is it is uh, it's carrying deoxygenated blood. Okay, I'm gonna write that down. Deoxy blood. And this goes to the lungs, so all of this from here goes to the lungs. Now from the lungs onwards, it becomes, you know, oxygenated because blood comes here. Oh, wait, yeah, this is wrong, not from the aorta. Don't write from aorta, that's wrong. From the lungs it comes to the pulmonary veins from all the sides. Actually, let me draw that. From all the sides it comes, and from there it will enter the left atrium. The left atrium, from there it will go to the left ventricle. From the left ventricle it will enter the aorta through the aortic valve, and from the aorta to the parts of the body. I know this is a bit chaotic, but I hope this makes you understand. So as you can see, there are mainly four valves here. The tricuspid valve is not actually tricuspid here, but remember, the right atrium and the right ventricle have tricuspid valves, left atrium, left ventricle have bicuspid valves between these. And along with that, you can see two other valves which are like half semi-lunar shaped. So these are called semi-lunar valves, and most of these are leaving the, I mean, they are towards the side where the ventricles meet the leaving this thing, which is pulmonary artery and aorta. This is pulmonary valve and aortic valve respectively. So let me write down the sequence here. What is this? Superior vena cava, superior vena cava, comma, inferior vena cava. Both of this gives into right atrium, from right atrium to right ventricle to pulmonary artery to lungs, from lungs to, okay, I am gonna take a U-turn because I mean here a turn, because this side is deoxy and this side is gonna be oxy. So this is from where oxygenated blood starts following, from lungs to pulmonary veins, from pulmonary veins to left atrium, from left atrium to left ventricle, from left ventricle to aorta, from aorta to body, other parts of the body. That is the sequence and the chain, as you can see. I'm gonna make a small note this thing here so that I make it easier for you guys to remember. A small note box. Let me add a box there. Okay, the first point of that, the first point you will remember is arteries are away, veins are towards. This even applies for pulmonary artery and pulmonary vein. The second point you're going to remember is all arteries carry oxygenated blood. Oxy blood. All veins carry deoxygenated blood. Deoxy blood. Except there's an exception everywhere. Pulmonary artery is deoxy. Pulmonary vein is oxy blood. Pulmonary means lungs, by the way, so I'm giving you tips to remember, okay? Oh, and this pinkish part is known as the septum. That's a wall. Why is it thick? I'm gonna write that down here. Why is the septum is a thick wall that goes towards the lining, right? Septum is this wall that differentiates, or this is not the question, but I'm gonna tell you another question that's required. Septum is the wall that divides the auricles and the ventricles from each other, okay? Oh yeah, here again, back. Semilunar valves, valves are aortic pulmonary, yeah, and continuation. Let me write that down. Try. They are called mitral valves. Mitral valves. So tricuspid, right? It's between right auricle and right ventricle. Bicuspid, left auricle and left ventricle. So if you ever get confused in semilunar valves, aortic and pulmonary, there is something I can tell you for you to remember. You have to just look at where the aorta and pulmonary arteries are, okay? And you'll figure it out. It's very easy. Along with that, we'll talk about systoles and diastoles. Systole, diastole. When it is in contraction, it is systole. When something is in relaxation, it's diastole. So during ventricular systole, it is auricular diastole and vice versa. There's still so much. Okay, so something they might ask is why um, why the walls towards the left are thicker. This is because And I'm gonna write a small answer here, but I'm gonna explain it. This is because left ventricles pump blood to the whole body, and hence there's a lot of energy required because um, it has to travel through the whole length of the body. Now that is done, I mean, this must be the easiest part. Let's talk about oh no, it's still not done. uh, let's talk about another thing is there is lub dub sound. Lub dub, lub dub, lub dub. Let me box that up first. Box it. This is all actually very important, so I would recommend you guys to go through it. I'll write it down here too. From examination point of view, very important. I'm gonna do that so that you guys go through that. What did happen? I got that. Very important from examination point of view. Go through this box which I've done. So the lub dub sound is caused when closing of lub is closing of cuspid valves, the tricuspid and bicuspid valves, atrioventricular valves. They are called atrioventricular valves. What is it? Mitral valves. They are mitral valves. Lub dub is mitral valve. Dub is semilunar valves. I mean, if you go through the notes without just studying or at least looking at the whatever I'm teaching right now, it might be difficult, but then if you studied, it's easier. I mean, you will understand it at the first sight of it. Let me put in another heart diagram once again, and I'll talk about the natural pacemaker in a second. Let's talk about this part, which pacemaking. So when your um, heart, I mean, is pumping, there is something that is shocking it. Okay, don't get scared. Something that's shocking it to provide it. So it starts off with this. There's a part that this is called the sinoatrial node. Okay, and it is connected in all parts of the right atrium. It goes like behind, and then behind this part, and then it enters the septum here, where it divides into multiple fibers like that. Okay, multiple fibers like that, and there is a small bundle here which does that. And just before this part leaves this thing, it forms another node there. Let me explain you all of this now. Yes. Okay. So this bacha right here is this SA node, sinoatrial node. The bacha following it is atrioventricular node. I have to write it down. Sure, don't. Atrioventricular node. This part here is known as bundle of his, and this ending part where all these fibers are coming off are Purkinje fibers. So the sequence just kind of goes like this. It's very easy. Sinoatrial node to atrioventricular node, from atrioventricular node to bundle of his BOH, from bundle of his to Purkinje fibers. That was kind of very easy. Hopefully you understood that. Now that we're done with most of it, how much notes have I made? Yay. Let me decrease the size back to 6. Let's go back to white. The pretty much last part of it, a few more stuff is there about this. Oh, I'll explain you in brief this part, the next upcoming parts in brief, and then there is a few circulations that we have to go through and live, and we are done. So let me start off with arteries the blood vessels. The blood vessels. So of course we have arteries. I'm gonna do this diagram so you don't have to just go through your notes here. I am bad at drawing, very bad. Arteries. And then the following is veins. Yeah. So each of these have 3 layers, and then you have to draw RBCs coming off RBCs. So let's start. Let me explain you all the layers and let's come down. The outermost layer. I hate my life. So the outermost layer here in both of these is connective tissue. The second most inner layer, tunica externa. These are also called tunica externa. Tunica media. Smooth muscles, aren't they? Yes, these are smooth muscles. And tunica interna. This one is endothelium. The end part and the hole in between is called the lumen. You can tell it, but this has a narrow lumen, wide lumen, thick muscles, thinner muscles, all that stuff. And then these are veins after. These have valves. Valves. This is veins. Then let's talk about capillaries. Capillaries are single cell and they have no muscles. That is the only thing you have to remember. This is for gaseous transfer. If they ask you to write, capillaries also have the property or power of dilating, so vasodilation and vasoconstriction. Uh, Something you have to remember is they're gonna ask you a sequence from the arteries to the veins, so it starts from artery branches into arterioles. From arterioles it goes to capillary. From capillary it is to venule. From venule it is the vein. That's the small sequence you have to. Now let's talk about circulations. First of all, the heart. Let's just consider this as a four-chambered heart. The lungs are here. There is two types of circulations which I'm gonna mark. Wait a second. Purple one circulation and a pink circulation. This is kind of divides into capillaries here. Okay. This lungs fella is pulmonary circulation. This pink one is systemic circulation. There is also another thing you have to remember is the hepatic portal vein. Let's talk about the hepatic portal vein. Starts from capillaries and it ends at capillaries. This is called double circulation, by the way. I forgot to add that on. There's not much going about that. So technically in the hepatic portal vein what happens is instead of all going and draining into this thing, all absorbed food is brought to the liver. I mean, the veins are taken to the liver. I'll show you. The blood is carried to the liver. So let's just say from the aorta, this is the heart. From the aorta, all the arteries are coming down and they go to the stomach. The stomach is this network here. There are three networks that are forming. This one is this large intestine and a small intestine network. All of these, instead of All of these join at one place. This place is then carried to liver. From there onwards, it is then taken to heart. Let me write down the places here: liver, stomach, uh, intestine, large, small intestine. This is hepatic portal. This is hepatic vein. There is, yeah. So it starts with this thing and it ends with the capillaries. The capillaries at lungs, and yeah. So what happens is the liver kind of detoxifies this thing there. So all the blood. So in the liver, it is deoxygenated blood. Deoxygenated but pure blood because it is kind of detoxified there. Sorry for my scribbling because I want to finish this fast. Now the next topic is pulse. This is just a kind of definition that you have to remember. Alternate expansion and elastic recoil of the wall of artery. This just might come as a definition, so read it through. Another this thing is blood pressure, BP. I'm gonna write that. The pressure exerted by blood on artery walls. If I want to highlight pressure exerted, blood, artery walls. There. Now let's talk about tissue fluid and lymph. So tissue fluid is this intercellular fluid, okay? It is just present between, when the plasma oozes out from the body, it is called tissue fluid. I don't think I have to explain that much, or I'll just write down. Plasma oozing out of the body. Plasma oozes out of plasma oozes out of the capillaries or blood vessels. That is when it is called tissue fluid. Lymph. So when this same tissue fluid is it re-enters the lymph channel or lymph vessel, it is called lymph. Okay, there's nothing different in it. It's just when it flows in the lymph channel, it is called lymph. It's called lymph. So lymph is when it drains into nodes called lymph nodes. pure lymph is created. Technically, but fresh lymph, not pure lymph, fresh lymph is created again. So it's technically kind of a recycling system. Now, lymph is composed of WBCs, water, some other solid matters. Technically, in solid matters, in sense, proteins and fats, and its functions. Yay! Functions of lymph. What are its functions? Well, I would recommend you to remember two. So, defense, because it mainly consists of WBCs, some linkage there, and another function can be drainage, E, drain away excess tissue fluid. It drains away excess tissue fluid. That's what it does. So, something to remember, it's just written here, but I think it might come as a one marker. Tonsils on the side of the neck are also lymph glands, so they come under the lymph system. Spleen is, there's this last part which is spleen. Let me write that down anyway. Reddish brown. It is a reddish brown organ, and organ which is behind this stomach, above the left of the kidney. You can remember this. Its functions are, it acts as a blood reservoir, so it stores blood, and in case of an accident or something like that, it kind of pours blood into your system where blood is worn out. It destroys old slash damaged RBCs, and in embryos it produces RBC. That's it. That is the end of the chapter. After it, RBCs. Yay! End of the chapter. The notes, I will try to give it. Let's see what happens. Okay, bye. I mean, that's the end of the hard chapter. Go have fun. We're done. Over. End. Khatam.